Science often begins when we realize the world is much bigger, smaller, older, faster, or slower than it looks.
Yeah, that incredible moment you realize your own human senses are really only giving you a tiny fraction of the whole story. I mean, you look at a forest and you think you see everything going on, right? But you're completely blind to these microscopic battles inside the leaves or the slow motion changes happening over decades.
Exactly. And to help unpack that entire hidden story, we want to welcome you specifically, our seventh grade life science students and your teachers and parents, to this Bioforge Educator Lab overview. Today, we are taking a deep dive into cross-cutting concept number 3, which is scale, proportion, and quantity.
Right, and for this dive, we're pulling directly from the Bioforge Life Science curriculum, plus a pretty massive stack of field research. But before we get into the concepts, I want you to really picture the setting for this conversation. We're not in some sterile, white, brightly lit classroom. I want you to imagine an Alaskan field station.
Oh, I love that vibe.
Right, so picture this sturdy wooden lab bench in front of us. It's covered in well-worn field journals, measuring tapes, scale bars, and a row of high-powered microscopes. And outside the window, there's mud, there's rain, and there's a massive, complex ecosystem operating on a dozen different levels all at once.
That is the perfect setup. So our mission today is to understand how size, time, distance, amount, and ratio fundamentally alter what scientists actually observe in nested biological and Earth systems. Because in science, just looking at something and using words like bigger, smaller, faster, or more, well, that simply isn't enough.
No, it's not.
Yeah,
Those words are entirely relative. I mean, what is fast to a glacier is incredibly slow to a hummingbird.
Yeah.
To actually make sense of the natural world, those concepts require concrete evidence. They require precise measurement. We need a specific toolkit to kind of deconstruct the physical world around us.
And that toolkit consists of scale, proportion, and quantity. So let's lay those tools out on our lab bench and define them. I'll take a crack at it, and you tell me if I'm on the right track.
Go for it.
Okay, first we have scale. I like to think of scale as the ultimate zoom lens. It's the magnitude of size, the magnitude of a time span, or the amount of energy you're looking at. So in biology, scale can zoom all the way in to a nanometer-wide strand of DNA, or it can zoom all the way out to map a caribou migration route covering thousands of kilometers.
Yeah, that's a great working definition. You change the scale, you change the scope of what you're observing. And then the second tool is proportion, which is the mathematical relationship between different dimensions or quantities. It's how things relate to each other within a specific system.
Like how parts of a whole compare.
Exactly. A classic example we use in biology is the surface area to volume ratio of a cell. That specific mathematical ratio dictates how easily nutrients can pass through the cell's outer membrane to feed the inside.
Okay, that makes sense. And the third tool is quantity. This one feels the most straightforward, honestly. Quantity is the numerical measurement in standardized units. It's the hard data, the actual numbers that we need to establish a baseline so we can spot patterns. You can't say an insect population is shrinking unless you have the quantity of bugs from last year compared to this year.
You got the toolkit down perfectly.
Well, here's where I want to push back a little, or at least challenge a really common misconception I think a lot of us have. When we talk about scale, why do things have to get so complicated mathematically?
What do you mean?
Let me use an analogy. Let's say I have a recipe for chicken soup that feeds 4 people. If I want to feed 40 people, I just multiply all the ingredients by 10. I scale it up linearly. Why can't we do that in biology? Like you watch sci-fi movies where a spider is blown up to the size of a brown bear. Why can't a bear-sized spider just exist in nature? It feels like we should be able to multiply the spider's blueprint by 1,000.
Oh man, I love this soup recipe analogy. But it highlights exactly why biology is so fascinating. Biological systems do not scale linearly, like a recipe. They exhibit nonlinear behavior.
Meaning the math doesn't just go up in a straight, predictable line.
Right. The physical laws that govern a system actually change depending on the scale you're operating at. Let's use a different analogy to explain your giant spider. Imagine you have a blueprint for a small, one-story wooden shed. If you take that exact blueprint and just multiply all the dimensions by 100 to build a skyscraper, what happens?
The wood splinters, the whole thing just collapses under its own weight because 2x4s can't hold up a 90-story building. You would need steel beams. The materials don't scale with the size.
That's exactly what happens to your giant spider. At the microscopic cellular level, gravity barely even matters. The dominant forces are viscous forces and molecular diffusion. For a tiny cell, moving through water is like you trying to wade through thick molasses. And tiny insects, well, they breathe through passive diffusion. Oxygen just naturally drifts through tiny tubes in their exoskeleton.
Wait, but if you blow that insect up to the size of a bear,
Gravity suddenly rules the system.
so the insect's exoskeleton would just shatter under its own massive weight, just like the wooden shed.
Yes. Plus, its internal volume, the sheer amount of tissue inside the giant bug, increases drastically faster than its surface area. It would instantly suffocate because it wouldn't have enough skin surface area to absorb the oxygen its massive new internal volume requires. A system's internal proportions shift fundamentally as its size changes.
Wow, that completely changes how I look at animals. Changing the scale changes the actual physical rules of the game, and we see this play out in nested systems all over the Alaskan wild.
Oh, absolutely.
And just real quick for our students listening, nested systems just mean that tiny systems live inside larger systems, which live inside even larger systems. Cells make up tissues, tissues make organisms, organisms make populations, and populations make ecosystems.
So let's look at a concrete example of this nested survival from the Bioforge materials. Surviving in Alaskan winter. If you look at an ecosystem in January, survival looks like, thick fur or migrating south. But really, macroscopic survival is dictated completely at the cellular scale.
Right, I'm thinking of the tundra lichen. It survives brutal freeze-thaw cycles that would turn, like, a tomato plant into absolute mush.
Exactly. And if we zoom into the microscopic scale, we find the mechanism for that. Lichen have extremely thick cell walls. When the water inside a normal plant cell freezes, the ice expands and literally bursts the cell wall, killing it.
Mhm.
But the lichen's microscopic architecture physically prevents that rupture.
Which is incredible, but my favorite example of this has to be the freeze-tolerant wood frog. Because if you look at this frog at the macroscopic organismal level in the middle of winter, it appears completely dead. It's frozen solid like a rock, no heartbeat, no brain activity at all.
But spring comes, it thaws out and hops away.
Yeah.
And the reason it survives is all about microchemistry. When water freezes, it forms these jagged ice crystals. Inside a cell, those ice crystals act like tiny daggers, just piercing the cell membrane. But the wood frog floods its cells with glucose, which is basic sugar.
So the sugar protects it.
Yeah, that glucose acts as a biological antifreeze. It lowers the freezing point of the liquid inside the cells, forcing the jagged ice crystals to form only in the spaces between the cells, rather than inside them. So the frog's macro survival is entirely dependent on this invisible microscopic mechanism.
We just saw how a tiny micro-level adaptation saves an organism. But scale works the other way too, right? Like a microscopic disruption can snowball into a massive, catastrophic landscape change. I'm thinking about the spruce bark beetle in south central and interior Alaska.
Oh, the spruce bark beetle is the textbook example of cross-scale disruption. These tiny beetles, they're about the size of a grain of rice, and they're actually native to Alaska. Historically, their populations were kept in check by deep, cold winters that would kill off a large quantity of the larvae.
But then we started seeing these microscopic incremental shifts in average winter climate temperatures. It doesn't feel like much to a human, but it's enough to keep the larvae from freezing.
Exactly. And because they survive the winter, their population math goes from linear to exponential. One beetle doesn't just replace itself with one offspring. One beetle creates 100, and those 100 create 10,000.
And then those millions of tiny insects bore into the spruce trees, specifically targeting the phloem, right? The microscopic transport tissue.
Yeah, your tree's internal plumbing that carries all the nutrients.
So the beetles essentially cut the nutrient highways. Millions of tiny beetles starving individual trees eventually scales up to fundamentally alter the macroscopic structure of entire forests. We are talking about millions of acres of dead gray trees. An entire ecosystem shifts, fire risks skyrocket, and animal habitats disappear, all because of a slight temperature change that favored a beetle you can barely see.
It's an incredible chain reaction. But let's trace another one of these nested systems, this time looking at how scale can build and sustain an ecosystem rather than disrupt it. Let's look at the gravel-to-gravel journey of the Pacific salmon.
Yes, the Pacific salmon life cycle is one of the most profound examples of scale, proportion, and quantity in biology. The sources define them as anadromous, meaning they hatch in freshwater rivers, migrate out to the saltwater ocean to grow massive, and then return to the exact same freshwater river to reproduce. Let's start at the micro scale, right at the beginning. Late fall, a female salmon creates a redd, which is a nest she digs in the river gravel.
Right, and at this stage, survival's a matter of microscopic proportions. Salmon embryos are buried in that gravel. For them to survive, The tiny, millimeter-wide spaces between the gravel pebbles must remain clear of fine mud and silt.
Because if silt fills those tiny gaps, the river water can't flow through the gravel, and the embryos need that cold, rushing water to bring them oxygen.
Exactly. If the microscopic gaps clog, the flow stops and the embryos suffocate. So the earliest stage of a 40-pound animal's life depends entirely on millimeter-scale water dynamics.
Then we scale way, way up, the macro scale. Those surviving fish hatch, grow, and migrate to the open ocean. They travel thousands of kilometers. And out in the ocean, they're bioaccumulating marine nutrients. They're eating and storing enormous quantities of marine-derived nitrogen and phosphorus in their bodies. They acquire up to 99% of their body mass in the ocean. Then, years later, they swim upstream, spawn, and die in the forest.
Which brings us to ecosystem proportion. This part is just amazing. These salmon are acting as a massive nutrient pump. Predators like brown bears and eagles catch the returning salmon and drag the carcasses deep into the forest.
And then the marine nutrients from the salmon decompose and act as this high-grade fertilizer for the terrestrial soil. We actually know this because scientists can track specific marine nitrogen isotopes inside the wood of the trees. The sources show that trees growing along salmon streams can grow up to 3 times faster than trees in areas without salmon.
A forest growing 3 times faster because of fish.
Yeah.
That is a massive proportional shift in the ecosystem's energy and matter cycle, all driven by the quantity of salmon returning from the ocean.
But let's talk about the math of scale here, because the sources brought up a really alarming quantitative threshold regarding those returning salmon. It has to do with nonlinear scaling again.
Yes, let's dive into the mechanics of this. We need to understand how length relates to volume. Volume cubes when length is linear.
Let's use a tangible analogy for you listening. Imagine you're building a cube out of small wooden blocks. If you build a cube that is 2 blocks long, 2 blocks wide, and 2 blocks high, you use 8 total blocks. But if you just increase the length of the sides by 1 single block, so it's 3 by 3 by 3, you don't just add a few blocks, you suddenly need 27 blocks. A small increase in length creates a massive explosion in total volume.
That is the perfect way to visualize it. Now apply that block math to a living salmon. If a female salmon returns to spawn, but because of environmental stress or, say, warmer ocean waters, she is just a little bit shorter than historical averages.
She doesn't just lose a little bit of internal space. Just like the blocks, her volume shrinks exponentially.
Exactly. The data shows that a small climate-driven drop in a returning salmon's length, just a 5% to 7% decrease in how long the fish is, results in a massive 24% to 35% decline in her total egg mass.
A 5% drop in length means up to a 35% drop in eggs. That is terrifying. It just goes to show that a tiny change you can measure with a ruler can completely wipe out a whole generation of fish. The math isn't just numbers on a page, it's a matter of population survival. It is why scientists must measure quantity so precisely.
Which naturally brings us to the history of science. Because, human eyes have a strict biological limit. We can only see things larger than about 0.1 millimeters. That's roughly the width of a human hair. Anything smaller than that is entirely invisible to us.
So scientific explanations are completely bound by the tools we invent. The history of science is really just the history of tools allowing us to see new scales.
Oh, 100%. Before the microscope, humans had absolutely no idea cells existed. It wasn't until microscope lenses were refined that scientists like Schwann and Schleiden could actually peer into that hidden world, see cells dividing, and build the foundation of cell theory. The tool unlocked the scale. The telescope did the exact same thing for astronomical expanses, proving we aren't the center of the universe.
And we use tools to understand the scale of time, too. Think about the geological record. By tracking the number of structural segments on trilobite fossils across deep, stacked layers of rock strata, scientists were able to understand deep time.
Right, the concept that the Earth isn't just thousands of years old, but billions of years old, allowing enough time for slow evolution to occur.
And we are still inventing new tools to observe new scales today, particularly in modern Alaskan conservation. Have you looked at the otolith thermal marking research in the Bioforge materials?
I have, and it is fascinating. So an otolith is a fish's tiny ear bone. And just like a tree grows rings every year, a fish's ear bone grows a microscopic layer of calcium every single day.
Hatchery managers use this biological mechanism to their advantage. They can actually manipulate the water temperature in the salmon hatchery, making it slightly colder for a few days. That mild temperature stress changes how the calcium forms, creating distinct microscopic dark rings on that tiny ear bone.
They literally create a barcode on a bone the size of a grain of sand. So when that fish is caught years later in the ocean, a scientist can put the ear bone under a microscope, read the barcode, and know exactly which river and which hatchery that specific fish came from.
That is so cool. But even wilder than reading ear bones is eDNA, environmental DNA. This tool completely changes the spatial scale of observation. As animals move through a watershed, they shed microscopic crumbs of themselves: skin cells, scales, mucus, waste.
So scientists don't even need to find the animal anymore. They can take a single cup of river water, run it through genetic filters in a lab, and extract those invisible traces of DNA. We can know a secretive or invasive species is hiding in a massive lake without ever actually seeing the animal.
But sometimes a massive visual change occurs on a landscape scale, and we have to use these tools to figure out the microscopic cause. I'm thinking about the recent mystery in the Brooks Range. We have these pristine, crystal clear wilderness rivers that suddenly turn bright, toxic orange.
Yeah, that was wild. To solve that mystery required scientists to use cross-scale reasoning. They had to look at the decadal scale of time, long-term climate warming. That warming thawed the permafrost, which is the layer of ground that usually stays frozen year-round. When that permafrost thawed, it acted like a broken freezer. It exposed a mineral in the soil called pyrite to oxygen and water.
Pyrite is commonly known as fool's gold, right? And at the microscopic scale, when pyrite hits oxygen and water, it undergoes chemical weathering. It essentially rusts, releasing iron and acid into the soil.
Exactly. That microscopic chemical reaction trickled into the groundwater, drained into tributaries, and scaled up to poison entire 174,000-hectare watersheds. The macro event, a giant orange river, was just the visible symptom of a microscopic landscape-wide chemical shift.
Now, when we talk about observing the natural world, we must remember that it isn't just about having the most expensive genetic filter or electron microscope. Observation takes many forms. And this brings us to the Alaska Cultural Standards, which are deeply integrated into the Bioforge curriculum. Scientific observation is also about generational presence.
This is a crucial point. For thousands of years, many Alaska Native communities have understood scale through careful, long-term, systemic observation. We are talking about generations of people living within an ecosystem, intimately tracking changing seasons, animal populations, weather patterns, sea ice thickness, tides, and harvest timing.
Right. Indigenous knowledge deeply integrates the concepts of proportion and quantity through practical daily management. It's the complex knowledge of how much is enough when harvesting a resource, when a plant is ready to be gathered, when sea ice conditions are safe based on subtle, proportional changes in wind and currents, and most importantly, when the ecological balance is shifting.
Modern ecology often relies on a grant that might fund, 3 to 5 years of study? Indigenous knowledge systems represent centuries of continuous data collection on stability and change within those nested systems.
And I really want to emphasize to our students listening, this cultural knowledge is relational. It is generational and it is highly practical. It is the intellectual property of those communities. It should never be treated as just a neat story or mere decoration in a science textbook. And it must never be used without proper context and permission.
Well said. It is rigorous, survival-tested observation that must be respected as foundational science.
So how do we put all of this theory into action? If you are a 7th grade student working through this Bioforge curriculum, how do you practice translating abstract mathematical proportions back into biological reality?
You do it by physically mapping the math. The curriculum includes an activity called the 50-meter microbe.
Oh, I love the mechanics of this one. Earlier we mentioned the limit of human sight is about 0.1 millimeters, the width of a human hair. In this activity, students take that single human hair and mathematically scale it up so that it is 50 meters wide. They go out to the school playground and use sidewalk chalk to draw a 50-meter wide hair.
Right, and that physical drawing forces the students to calculate and draw proportional models of other microscopic things next to it. If a human hair is 50 meters wide, they have to use proportional formulas to figure out how big an animal cell is. How big is a bacterium? How big is a virus?
And when they actually draw it out, the animal cell is the size of a large room, but the virus is just the size of a small pebble. It visually proves why a single animal cell can get infected by and hold thousands and thousands of tiny viral particles before it bursts. It makes the invisible math completely tangible.
And students then pair that playground activity with virtual tools. They use digital interactive sliders on their tablets to navigate across 10 orders of magnitude. They can zoom all the way from a coffee bean down to a carbon atom.
And as they zoom, they map out what can be seen with a standard classroom light microscope versus what requires an incredibly powerful electron microscope. It teaches them the hard limitations of their tools.
It also prepares them for complex ecosystem math, like calculating trophic level energy loss. In the curriculum, they learn the 10% rule.
Let's break that rule down. A trophic level is basically just a step in the food chain. So a plant absorbs sunlight and makes 100 units of energy. A moose eats the plant. But the moose doesn't store 100 units of energy in its meat. It burns 90 units just walking around and staying warm in the winter. It only stores 10 units.
Yeah, and then a wolf eats the moose, and again, 90% of that energy is lost as heat. The wolf only gets 1 unit of the original solar energy.
That proportion, that massive energy loss at every step, is the quantitative reason why an ecosystem can support millions of plants, thousands of herbivores, but only a very small handful of apex predators. Proportion dictates the entire shape of the food web.
So measurement and proportion are really your ultimate guides. They're the only way to truly understand the mechanics of the natural world, for the ice crystals in a wood frog's veins to the salmon feeding a forest.
Changing the scale changes what you can see.
It changes the physical rules, the patterns, and the very questions we know to ask.
Exactly. So we want to leave you with a thought to mull over. Take out your field journal or discuss this in your classroom today. Finish this sentence. One thing I understand differently when I zoom in or zoom out is